So this is the one that I made a screenshot of that you are provided in your folder. Looks like that. You get the blobs. We have everything in there. We have some basic pre-processing. Here this median. Then you have auto thresholding, and then nothing else. Then we have a simple detection of ROI, measuring the areas directly in the same node. And then some visualization. I don't have here the set overlay. It just renders out the RGB finally. But you have seen how to do this. So, and how you will come up now with these pipelines when you are confronted with an image analysis tasks. Well, that's why I showed you yesterday this incomplete toolbox of many things, but it's a good start. And I also took all of the toolbox algorithms that I showcased and then made an example in JIPipe that you can just run. That contains basically all the algorithms that are in the toolbox that you can just run from within JIPipe, and you can see how they behave on the data. But I will showcase here in the presentation a quick overview of the equivalent of what I explained in the toolbox or in ImageJ in JIPipe. So you have the things that you are familiar with already, how to get files into JIPipe with this file list that you can just drag and drop. You can also create it manually via this add node search panel and then manually add one or multiple paths into it. There's a sister node for folders. So if you give it a directory and drop a directory in, it will automatically make a folder list. If you have like a mix of paths and directories, then it will actually make it's called a path list because it doesn't know which is a directory and which is a file. Then you have seen already the import image node that imports images, and it by default attempts to use ImageJ standard importer. This is the same one that if you go to ImageJ file open, it for example imports PNG, TIFF, and others. But if you confront this import image one with something that is a bit more advanced file format wise, like a CZI file or an OME TIFF, it will actually internally forward it to the bioformats importer. You will see like a little note in the log. Usually there's things that we did were very fast, but in the log you can find information what JIPipe is currently doing. And there it usually will say, okay, I'm using bioformats importer because this is a file that ImageJ cannot handle. If you want more control over your bioformats things, there is a dedicated bioformats importer, which also grabs files, but then outputs a so-called OME image. This is basically something like an image, but it also has some information in there that comes from bioformats, like the metadata that is stored inside the file. Bioformats is very, very strict in standard for how it saves metadata and what it extracts. And this kind of image here, it has this metadata in there, and you can actually use it in your pipeline to grab information out, use it in your analysis. Then the pre-processing algorithms, how they are called in JIPipe. We have already used the median filter 2D to get a simple median filtering. It's exactly the same one as ImageJ does. There's also a Gaussian blur 2D variant, because there's also a 3D. And for histogram enhancement, in ImageJ it's in process contrast enhancement. In JIPipe it's called the histogram-based contrast enhancer. And there you have both the histogram stretching and equalization, and then some more settings that are also available there. They're all the same as in ImageJ. We have a Sobel edge detector and also a basic Sobel filter, and also a Laplacian filter for some basic pre-processing and feature enhancement. Then Kani edge detector is also available. It's named Kani edge detector 2D. It makes these nice lines that are already binarized. And you also get a Laplacian of Gaussian and difference of Gaussian in JIPipe as well. Just be careful that this one here will make a 32-bit image because it can go into the negative space. And one of the exercise pipelines that I provide you, this actually does a conversion, the necessary conversion where it basically takes only the positive part. But it is explained in there. So if you choose to use this example. Then the morphological operations, they are all under the one umbrella called morphological operation 2D. And there you will find everything that I showed you: dilation, erosion, opening, closing, and the gradient operation. There's a parameter setting in there where you can just choose. And then also the element, if it's like a disc-shaped element, for example, if you want to have more rounder objects, then a disc is good. And then you can also set the radius. And you can also add a border around it, because morphological operations are sometimes a bit weird when with image borders, because mathematically it's not really defined what's happening outside, so they usually do something. And there you can actually determine what should happen there. So then also a common operation from ImageJ is in some way masking. So you want to delete maybe pixels that are in a specific area. You have a mask or you have a region of interest, and then you want to set the values in your image to 0 inside this mask, outside this mask, or inside the ROI or outside the ROI. And JIPipe also has it. It's called set to value grayscale for grayscale images. And there's also a set to value color for color images. And there you can configure in the settings if it should receive a mask or a ROI, and if it's inside or outside. Let me show it to you. And it's called set to value. You see here, when you search set to value, it will hit multiple things here. These 5. This is the original node, and then you have a few pre-configured ones. So you don't have to go and then configure it. For example, I want to set the value inside my region of interest to something. I use that one. It's pre-configured. If I go to the parameters, it says here only apply to inside ROI, and then I can give the value that it should set to. For example, I want to set it to 0. It's used for post-processing. This is the equivalent of an ImageJ if you use a ROI or you select something and then set it to something else. In JIPipe, you can't do this interactively because it's an automated language. So here this is the equivalent of it. So then, so far we have used ROI-based measurements using fine particles 2D. Also the ImageJ particle finder. This is straightforward. If you're coming from ImageJ, the standard stuff. So ROIs in ImageJ are contours around areas. These ROIs are super low memory, they are super fast, and have one disadvantage. They cannot have holes. You cannot draw something around something that has a hole. But you were able to make some in ImageJ that have holes. And this is possible because ImageJ has some, they're called composite ROIs, where basically you have multiple ROIs stacked into each other. And then they can work around that issue to basically introduce holes artificially. I would not rely on those because some of these, some ImageJ algorithms absolutely do not like these composite ROIs, and then they do something, and then you get weird results out of those. So a safer choice for anything that has holes is label-based analysis. So when you have a mask, you use a different node to convert them into a label image. It's called Connected Components Labeling 2D. It comes from MorphoLibJ originally. These are slower. They need more memory because they are full images, but they can be basically any shape. You can have them nested inside. You can basically do anything with them. And JIPipe has many equivalent algorithms for labels versus ROIs. So you usually find something that extracts statistics from a region of interest as a dedicated node. Then you find the same thing also for labels here. You see here this extract label statistics. This is basically the main statistics extractor. While the fine particles 2D finds the measurements immediately. Just you need 2 steps here. But you get just a lot of benefits. So let's switch the data to something that's more interesting. And for that, I have chosen this kidney segmentation. So this is from lightsheet data. And specifically, we will only handle the 2D slices of this lightsheet data from a study where a new clearing method was developed. And in this study, it was important because the clearing shrinks the tissue size to find the tissue area, record that, and then also have the ability from a pipeline that counts the glomeruli inside this plane that you see. So these functional units of the kidney. And this pipeline that we will do now will do both. It will find both the tissue and the glomeruli inside. And we find the data in this kidney basic directory here. So there you find many different files. Don't open this kidney 3D 20 slices unless you want to see them in 3D. We start very simply with this kidney.tif image. Just no number, kidney as is. So, and then I showed you already importing everything. We can do this efficiently now. You can add the import image, but I want to show you something else as well. Instead of using the import image here, which you can definitively do. So this works, but JIPipe also has some nice tool that is sometimes useful for these. These data types here, they are not only for fun there and to guide you when you're building your pipeline. JIPipe can also help you when building your pipeline based on this. There's a function that's called find matching node. So if you have an output or an input of a type, you can ask basically JIPipe, okay, what fits to this input or output? You will get like a search box, and then this shows you only nodes that fit towards this given output as an input. And it will also find the import image because it eats a file and then converts it into an image, but also many other tools as well. And there you can click the add button, or you can click anywhere. And then you can not only add it to the graph, but also auto-connect it directly. So I'm going to click here this connect to files. Then it made me my connection, so I have less clicks to do. You can also call upon this feature by double-clicking it. So you can just double-click these outputs or inputs. It can adapt to it. Then it would then search something that makes this required input type, if you ask it for an input, and then adds it in. Or you can just use the panel if you are more familiar with it. So we have the tissue now in, it's loaded in, so we can look at it. And I can open it up. So this is how the kidney looks like. Has some tissue was quite easy to find. You can enhance the contrast a bit. Look inside, there are bright areas inside the tissue. And remember from yesterday, we used the specific operation to actually find them. And we will do this now also in JIPipe. And actually a lot of operations that I showed you yesterday all were shown on this data originally. So let's close that and start with some simple denoising. I personally just like always the median filter. So I added median filter 2D. Be careful, there are different variants of it that somehow match just the normal regular median filter 2D. Then we have some basic denoising going on. Yeah, it's fine like that. And then we are at the auto thresholding step directly after that. So auto threshold 2D, you should be familiar with that now. We have the basic workflow now. And then now we have to choose which thresholding works the best. So that's the default, and you see it's not finding everything perfectly. So you see here, this is quite far apart. So maybe we need a thresholding method that is a bit more sensitively towards what we need. And to help you with that, so you have seen maybe when you look through this auto threshold 2D, there's an option missing from ImageShade, this try all, that would help you with finding all these different thresholds. And you can do this in JIPipe, but it's not a dedicated node. But usually it's like a set of nodes that you can use to figure these out. And I actually made you one that you can just reuse. And it's available via a template. So you have maybe seen in the lecture data that I provided to you, there's this file here. auto threshold 2D try-all.json. It's here in this lecture data. And we are going to import this now as a so-called node template. I will explain this a bit later, how you make them on your own. This is part of another lecture. But let's import them into JIPipe. So let's click here into JIPipe, and then we go in, not into the Add Nodes panel, we go into the Templates panel, which is one below the Add Nodes. And then there, we just right-click somewhere, and then click Import from File. And then we navigate through our lecture data, and there is this auto threshold 2D try-all.json. And then we just open it up. And then it asks you globally or inside project. And you just tell globally, because then you have it in all projects. And you see that it should appear in this list. So and then this thing behaves similar to the node panel. You can just drag it in, or you can double click it. And then you get a node, because what this template system is basically something that is saved somewhere that you can just reuse. And I can just connect it to my median filter here. It says here this is the try all. You can even open these up and then see what's inside. I just show it, but we will take care of that later on, what is actually going on there, because it uses some advanced features. And then we can run it. And it will not only make a montage, but also it gives us all the thresholds results. So you can, it has two outputs. Thresholded contains all the results that it generated, all the different thresholding methods. And then also a montage just as image.j. Yeah. And then you see here, and you can, just like an image, you can easily compare how well each thresholding algorithm behaves. Let's make it larger. So you see here some of these, they react more to the glomeruli. Some are very good for the tissue, or seem to be good for the tissue. If you are familiar with the image.j thresholding methods, usually the Huang threshold is very good if you have something that is quite faint, because it reacts very, very strongly towards objects. And then you get now these thresholded results, and you see it, you get one for each threshold. It's in this other output, in this thresholded output. So here they are as is. And then you can just open them up. And this is why there's this JIPipe viewer. Here you can just browse to them. You can just go here at the top left, next, browse to them in JIPipe directly. And then how do I know which thresholding it is? You can open this panels here, it says annotations. And there it says what is the method, because this node will automatically add this information into the image, so you can read it out. And in this way, you can easily select, okay, which one looks the best? Ozu, percentile, no. Yen, okay, this is interesting, how it behaves. Shandong doesn't find anything. Intermodes, triangle, min error, no. Moments, no. Isodata, maybe, but yes, you have these parts that are cut apart. The other isodata, Renji, entropy, Li is good. It mostly behaves like Huang, but Huang usually works best for these. So let's set it now to Huang, because this one worked the best. And then when I run it, then boom. So we have a quite nice image, but we have here these holes in the middle. We have these border artifacts. They are not so strong under Huang. But we can smooth this out a bit, and then there are also some images where it doesn't work perfectly, and you get weird artifacts in the border. And then we have here these wrongly detected region on the outside as well. So we want to get rid of all of those. So let's do this with the methods that we've learned yesterday. We could use morphological closing for that, especially because these holes are super small. But what I know from the data that there are some that have like big holes in the middle. So let's not do this. Use instead the morphological hole filling, this one that uses flat filling. So in the add nodes, morphological hole filling 2D. It's in images morphology. If you want to go via right click, then it's here under morphology, morphological hole filling, 2D. But I found it already. And then it eats a mask, and then it produces a mask, as it says here in the, you see it in the icons. And then let's try it out what it does. So I open it up. So now it filled here the holes. So question is now, are we done? Because it looks good here for our test image, but I know a data set where it doesn't work. We can change the inputs very easily. I'm just dragging kidney 6 in. And then disconnecting that and connect it to the kidney. Now you can see something interesting happening here with how the workflow cache system works. So if I run now with update cache, only the hole filling, specifically update cache, not the intermediate one, then you will see that all the results here disappeared in between. You see there were results here. They are now gone because JIPipe noticed, oh wait, this one here is new. You changed something. So it basically invalidated all the results that were stored here for me. And it's smart. It knows when something changes and then will understand, okay, I have to clear this because it's making no sense, something changed. And it will rerun this then later on. And then it stored only here the morphological hole filling. When you see then with this data, that's why I wanted to show it to you, you see it already here in the picture, that our hole filling failed. This is quite inconvenient. And why it is, let's maybe check the original data. And that's why sometimes this cache intermediate result is super useful. If I run this one here with cache intermediate results, it's fine enough. It's relatively small data. You can go back and then investigate what's going on. You can browse through it. Okay, for this simple thing, it's easy. You can just do it in the pipeline. Later on, you can also trace it. So you get like a tracer where you can see how the data developed through the pipeline. But I will show it later on when we have something that is more interesting or multiple data. You see, if I go back, we have this cut, also all these parts here disconnected. Something that we have to account for. And this is why I explained this protocol where you apply the hole filling, but together with some morphological operations. Remember when you dilate it first, then you apply the hole filling, which then would close here, the dilation would close this here if you set it large enough, and also connect all of these things that are floating. Then we apply the hole filling, would close any holes that would be maybe in the inside, and then we deflate it again with an erosion. And then this way we have the original size still restored, but all the holes are closed. So let's implement this in JIPipe. So instead of directly doing the hole filling, I am disconnecting that one and making some space. So I can add a morphological operation inside there. And what we want is a dilation. So you should be able to search for dilation in the search box, and it will find the morphological operation 2D dilation, which is a pre-configured node that's just set to dilation. You can also just use the morphological operation 2D as is, and then you set it up. So we have this morphological operation 2D. Then we have to configure it. It's the same one as an image that you get from morpholib.j. Parameters, it's set already to dilation. And then for that we need a quite large radius. We're doing a dilation of 20. Then it inflates everything. Then we can do a hole filling after that. And then we do a erosion. So I can, of course, hunt down the node here again. Or I can just copy paste it, or I can duplicate it. So maybe make a bit of space. And then a nice key binding is Control D. If you go with your mouse where the node should be that you have highlighted, you click Control D, it will duplicate it there. Or you can just copy paste it normally. That doesn't matter in the end. But this one, we have to configure into erosion. And so you are not getting confused here. I name them dilation. This is the radius as 20. And then this is the erosion. And this also radius 20. So we get our original size back. And then let's run it with update cache. Then I can show you how it develops. We have the auto threshold 2D result. So we have the holes. Then we have the dilated variant. And you see it inflated everything, dilated the area, but you still have the holes inside and it still doesn't cover everything. And then remember that you can absolutely destroy the structure if you increase this radius too much. Just enough. Then we can apply the hole filling. Then, boom, all our holes are closed. But now this would yield skewed statistics regarding the tissue area, because this was an important statistic from this original data set. So we have to erode it again, so we get approximately the same area back. So let me connect again the kidney.tif instead of the kidney 6, so to see if it still works. And then if I run update cache here, you see how all these intermediate results disappeared because they are now invalid. And there we have our kidney holes are filled. We have still this weird artifact, but we're going to get rid of that very soon. So we have this one, original kidney. This is then our pipeline. And then you can just replug the file list if you want to try it on kidney 6. So, and then let's do the ROI conversion. Similar as before, in ImageJ, we'll find particles 2D. Don't forget there's a 3D variant as well. We don't want that. And then we find 7 ROIs this time. Okay, this one. Oh, look at these artifacts here at the top. Found them all as individual ROIs. But we will get rid of these individual ROIs now. Easiest way is just setting a size filter. So let's try the min particle size, similar as you would do in ImageJ. We can do like maybe 5,000. Then it should get rid of all these small artifacts in the size. There are also dedicated filters that remove things that are at the border, but we don't need this now. Min particles filter is absolutely enough. I write it down in the name so you don't forget it. We should have, if I rerun it, 1 ROI only. And then this one is the kidney ROI and nothing else. So perfect, works fine. And then we also get the measurements, because we also got the area in square pixels, I think, because it doesn't have the physical sizes attached here, this image. We are basically done for that. Let's visualize it. And let's do this with the simple one with convert ROI to RGB. So this can be later exported. And then, okay, this is a bit annoying if it's inverted, because my image here, I wanted to do it from the left side. So I can just right click it and then move it to the left, and then I can switch it around. Some nodes care about this order, but most don't. It's mostly nodes that merge something into like merge channels or something that cares about the order. And then I give it the original image as well. And then we can render it out. And then we have it, maybe we make the line a bit thicker. You can override all of these rendering behaviors per ROI. So there are dedicated nodes that you will see later. Or you can also do this directly here in these rendering nodes. For example, I can tell it, override my line width a bit, make it a bit larger. You can also make the color different. This default color here is yellow. If the ROI basically has no color set, then it will opt to yellow. But you can also force it to something. And you can also add opacity. This is something that ImageJ doesn't do. So then this way you have like a nice outline that can be later on saved when I show you actually how to save stuff out. I can quickly show how to get the glomeruli, but this is very straightforward to get the glomeruli. So now you can make use of something that JIPipe is very good at. So what you can do in JIPipe is very easily. For example, I have here my pre-processed image. Looks like that. But now we want to have the glomeruli instead. If you remember correctly, there were some thresholding methods that reacted well to it, but I show you how well they actually work. So the thresholding methods, they are all okay-ish. If you look from them from outside, from this dry oil, basically they are looking fine. But if you zoom in, then none of them actually is good. It misses a lot of these. And this is why we need a proper feature enhancement to really find the blobs out, because this is where the blobs example comes from. We need to enhance these blob-like features from the background so we can do the thresholding nicely. So let's, starting from the median filter, add another operation, which is the top head. So you can search it, top head, specifically a white top head. And then we are going to add this as a branch here. And we start off the median filter with that. And then I write you down what is the settings for this top head, because it basically needs to be configured in a way that it must fit towards the size of the objects that are in there. So the morphological operation 2D is a top head, white top head, and the radius needs to be approximately the size of the object that you're looking for, and then it will enhance these specifically and suppress the background down. Well, let's see if it worked. Let's do some contrast enhancement. Some of these thresholding methods definitely will hit that. And then we can do some basic auto thresholding. Auto threshold 2D. And the default method, I think, works the best in my test. Then looks good, I think. If I run it with the default auto threshold, don't configure anything. And then let's directly after that do a fine particles 2D. When I'm super lazy, I just copy that one. And I configure it with small particles should be allowed. Min particle size is 0. And the circularity should be larger than 0.7 here. So it found 276 boys. And then let's finally draw them on top. So I can draw my glomeruli that were detected on top of the original image. So to see what it does. Maybe I change the sizes a bit down, like 2, so they can be seen. It's somewhat okay. So there's actually some additional fitting that can be applied to make it a bit better. But this is good enough for some tests. There can be some additional enhancement to be added. And then we can also do something nice. Maybe we want also the glomeruli and the tissue both in one visualization as well. So I am duplicating this one here. This one I want to keep, but I also want to another visualization. So you can also duplicate that one. And then I am drawing my glomeruli not on the original image, but I can also draw it on the one that the tissue made, because it just yields an image that is out. So we can stack stuff on top of each other very easily. But here I want the glomeruli to be in a different color, so I can override the line color to maybe blue or something. It's all in the settings of the node. And this way we can make some nicely stacked visualizations. Let's see how it looks like, where you see the tissue and the glomeruli in the same visualization. One is yellow, one is blue. So maybe I can zoom out a bit. So you see the full pipeline. So we have here the white top head on the median filter, so the branching operation, auto threshold 2D with default, and then a fine particles where we leave the limits alone. But we say, okay, at least 0.7 circularity, otherwise it would find some artifact. It would find also these here. But then the circularity filter gets rid of that. And then we have our regions of interest that are already measured. And then we draw them onto the original image. But we also want to draw them as additional visualization on top of the image that we already visualized that has the tissue. And then we will handle the bit more advanced features that you will get from JIPipe. So this is our start here.